Let's begin our conversation of regulation by talking about the regulation of the transition step first. And of course, more expressly, that means the regulation of the huge pyruvate dehydrogenase complex, the multi-enzyme complex of E1, E2, and E3. And we know now that this is really the regulation that affects the contribution of acetyl-CoA coming from carbohydrate catabolism. So to remind ourselves of what's going on here, let's go ahead and jot that reaction down, recalling that the transition step is the step in which pyruvate is going to be converted to acetyl-CoA, and we'll just go ahead and get all the reactants written in here. So pyruvate plus coenzyme A plus NAD plus will then be converted by the pyruvate dehydrogenase complex I'm just going to abbreviate pyruvate dehydrogenase. That's supposed to be a D. And that's going to get converted into acetyl-CoA plus NADH Of course, don't forget the thoroughly worn out, totally used up CO2 that's lost there. Now, the reason I've gone ahead and written this out, because it turns out that the regulation of the pyruvate dehydrogenase complex is really quite simple, in that it is regulated by something called mass action effects. That is to say, in simpler terms, the products will inhibit, whereas the reactants will activate. Now, we don't find that terribly surprising at all, right? Reactants will be activating, feed-forward activating, products will be inhibiting. Sounds pretty much the same as what we've talked about before, but it gets a fancy term in this case as mass action effects, because we're recognizing that the ratio of products to reactants will matter. If products are high, then inhibition will occur. If reactants are high as compared to products, then activation will occur. So we're basically seeing this mass action effects. So let's go ahead and jot that down. So regulation of this complex controls the supply of acetyl-CoA from pyruvate and hence from carbohydrate degradation. We already mentioned that. So in mammalian cells and in E. coli, the E2, or dihydrolipoamide acetyltransferase, and dihydrolipoamide dehydrogenase are the two components of the multi-enzyme complex that are regulated by these mass action effects. So we recognize then that the substrates will activate and the products will inhibit. So E2, namely, or the dihydrolipoamide acetyltransferase, is going to be inhibited when acetyl-CoA as compared to coenzyme A is, okay, so remembering back, acetyl-CoA is a product, so it will inhibit. So if it's high, that's when it's going to be inhibited. So we'll say when it's high. And we can recognize E3 being inhibited likewise when we have high NADH product over lower reactant, NAD+. So we're recognizing those impacts of mass action ratios. Awesome. Pyruvate dehydrogenase is also regulated in another way, and we need to go on and talk about that more convoluted way next. Regulation of the pyruvate dehydrogenase complex just gets crazier and crazier, because in addition to E2 and E3 being regulated allosterically, we also see regulation of E1 in the form of covalent modification. So remember, that means the addition of a group covalently linked onto the enzyme and changing its activity. In this particular case, our E1 is actually inactivated by addition of a phosphoryl group. So the active pyruvate dehydrogenase is the dephosphorylated form. So when E1 is dephosphorylated, it is going to be popping out products. However, when you get the addition of a phosphoryl group by the hydrolysis of an ATP and the converter enzyme acting on E1, so E1 would be the interconvertible enzyme, the converter enzyme would act on that. It does also by transferring a phosphoryl group from ATP onto the E1 enzyme. So we know then that that enzyme must have to be a kinase, right? By default, we know that must be the name. Pyruvate dehydrogenase kinase is the very fitting name, or we could even just think of it as being E1 kinase, right? That is the enzyme. So upon being phosphorylated, E1 is inactivated. So this is another level of regulation. So in addition to E2 and E3 responding to either reactant accumulation or product accumulation, phosphorylation of E1 will downregulate the entire complex as well. But things don't stop there, and this is where it gets really deep in here, because it turns out that pyruvate dehydrogenase kinase is also allosterically regulated. So to think this through, what we have to realize is that if pyruvate dehydrogenase kinase is active, it's going to inactivate the E1. So the entire pyruvate dehydrogenase complex will be inactivated. So think this through one more time. Pyruvate dehydrogenase kinase, if it's active, then that means we're going to see inactive E1. If the kinase is inactivated, it means we're going to see activation of E1. So to think that through, remember how we said that products of the transition step are going to downregulate the enzymes 2, 3, E1, E2, E3, right? The products are going to downregulate. And so this, in this case, will be enacted through the kinase. So if we have accumulating products, we know that's going to downregulate or inactivate the pyruvate dehydrogenase E1. So it must activate the kinase. So things like NADH and acetyl-CoA products are going to activate that kinase. Hopefully that kind of makes sense. Now, on the flip side, we would recognize that accumulating reactants, right, we know is going to tend to activate the E1, and so we would see it inactivating or downregulating the kinase or inhibiting the kinase. So pyruvate, NAD+, coenzyme A, and ADP, these are all going to inhibit the kinase. Now, there's one there that's not a reactant, right? NAD+ is, coenzyme A is, and pyruvate is, but look at ADP getting its head in all the mess. Well, that's because ADP, if it's present at high levels, simply indicates a need for ATP, right? Up the ATP production. And so ADP will want to, in fact, inhibit the kinase to then likewise activate the dehydrogenase so that we get more production of acetyl-CoA, more and more carb breakdown to produce acetyl-CoA to fuel the TCA cycle, right? Okay, good. We're getting through the depth here. So now, to get really sexy-minded, remember this article that we looked at a little bit earlier in our coverage. Remember that we talked about how in cancerous cells, glycolytic pathways tend to be upregulated and aerobic respiration downregulated. Recall that we mentioned that one of the techniques for trying to target cancers is actually to try to upregulate pyruvate dehydrogenase complex. And the way in which this article reports doing that is actually by inhibiting the pyruvate dehydrogenase kinase, so that in fact that stops the phosphorylation and inactivation of pyruvate dehydrogenase, therefore upregulating the pyruvate dehydrogenase complex. Now we recognize that as is always true, there is another converter enzyme that can cleave off the phosphoryl group. We know this one won't be a kinase, instead it will be a phosphatase. It will cleave that phosphate group. So pyruvate dehydrogenase phosphatase will cleave off the phosphoryl group, leaving again the active pyruvate dehydrogenase form, or E1. So we'll look here at the fact that pyruvate, I know it just keeps going and going, pyruvate dehydrogenase phosphatase is also regulated, and it's regulated by calcium. And we know that that muscle contraction is stimulated by release of calcium 2+ ions. Now, there are some other things that lead to release of calcium 2+, but let's think of it in that way. There are some hormones, calcitonin and whatnot, that can lead to that up in calcium release. But if we think about it as a muscle contraction, calcium is upregulating the dehydrogenase or pyruvate dehydrogenase phosphatase. Okay, well that makes sense, because what that means is if pyruvate dehydrogenase phosphatase is upregulated, it cleaves the phosphoryl group off of E1, activating E1, activating the transition step, increasing the rate of the transition step. And that makes sense because with all that contracting muscle tissue, it indicates a need for energy production. So hopefully this helps you think through this very convoluted regulation. We're not quite done with regulation yet, because now that we've talked about the transition step and the pyruvate dehydrogenase complex, we need to now look at the 3 enzymes in the TCA cycle that are regulated as well, beginning with citrate synthase, of course, that catalyzes step 1. But remember how nebulous this particular step was, because remember that scientists are having trouble even measuring the accurate concentrations of oxaloacetate. And so we likewise might say that the mechanism of regulation at this step is also not very well established. So we'll go ahead and jump into the next step, and just to help us visualize all of this, I'm going to jot a quick skeleton of the TCA to remind us that regulation is taking place at citrate synthase, which is that first metabolically irreversible step. Then we have the near equilibrium step, that's step number 2, in which citrate is converted to isocitrate and isomerization. And then we have steps 3 and 4 that are metabolically irreversible. And those are the next 2 steps that are subject to regulation. And remember, those were catalyzed by isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. So let's first look at the regulation of isocitrate dehydrogenase, and then we can move into the regulation of alpha-ketoglutarate dehydrogenase. And once again, remember how it's a multi-enzyme complex, just like pyruvate dehydrogenase. So we could see in this case again the regulation at E1, E2, and E3, right? Oh man. So let's go ahead and start with isocitrate dehydrogenase, and note that it's activated by calcium, again contracting muscle tissue's need for energy, run that TCA cycle. It's also regulated by ADP, again indicating need to run the cycle. It's inhibited by NADH. Now we can make sense of that too, product inhibition, because remember that NADH is a product of the step catalyzed by isocitrate dehydrogenase. We can also make a little more sense of that by thinking about what happens if the NADH that's made in that step has nowhere to go. For example, if the ETC has slowed back and is running really crawling and slow, that NADH is going to accumulate. It's kind of like a traffic jam, right? The NADH just keep accumulating. And so that's going to signal to the TCA, whoa, slow down, hold, pull the reins, right? Slow down. So that's how that is enacted. In E. coli, we also see another level of regulation on isocitrate dehydrogenase covalent modification that's not as well established in other systems. So let's move to our multi-enzyme complex, our second multi-enzyme complex, alpha-ketoglutarate dehydrogenase. And it turns out that it too is regulated by calcium. Calcium binds to the E1 enzyme, and it actually, so this is kind of a fun one to think through. When calcium binds to E1, it actually literally changes the Km. So if we know that calcium activates in all of these cases, right, it stimulates and says need for energy, then we would expect that this would upregulate alpha-ketoglutarate dehydrogenase. So if it's going to upregulate alpha-ketoglutarate dehydrogenase by changing the Km, what would it do to the value of the Km, right? It would have to increase the apparent affinity for substrate or lower the Km, right? So it decreases the Km of alpha-ketoglutarate dehydrogenase. So this is one way of increasing the rate of that step and increasing the rate of formation of product. Some of the feedback inhibitions that you might think of are also there, because remember again, NADH is a product of that step, and as is succinyl-CoA, both are feedback inhibitors of alpha-ketoglutarate dehydrogenase. Now, this is interesting because this has literally only been proven in vitro and not in vivo. Now, in vitro literally means doing it on the bench top, right? Mad scientists doing it on the bench top. That paints a funny picture, but yeah, in vitro, right, on the bench top. In vivo means in the body, and so this hasn't really been shown in the body. So there are nuances here that maybe haven't been entirely worked out. But that says it for the regulation of isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. As we complete our coverage of the TCA cycle, we come back to where we started. That is, thinking about the cycle itself as the hub of aerobic metabolism and a cycle that can ebb and flow and flux and change. That is, intermediates can be added or intermediates can be siphoned away to change the speed of the overall cycle. I want to talk now about the idea that intermediates can be siphoned away from this pathway. And I think about those of you who have expressed a love of Legos. It's very important to note that a lot of the intermediates in the TCA cycle are Legos that can be utilized as building blocks in anabolic reactions. So in many cases, the fate of the intermediates will depend upon the need of the cell. If the cell needs more reducing power in ATP, then it will continue to catabolize these intermediates. But if there is some need for Legos to build larger molecules in anabolism, then these intermediates will be siphoned away. And I want to give you just some examples of the way in which these might be used as precursor metabolites, and we'll begin over with our first intermediate or last intermediate, depending on how you look at it, oxaloacetate. And it's worthy of note, we're going to talk in a minute about a reaction that can fill up oxaloacetate, but there are reactions that can also pull away oxaloacetate, and sometimes they're one and the same. For example, oxaloacetate can actually get converted into pyruvate, and pyruvate, if glycolysis is turned backwards and gluconeogenesis is run, then that can be used to synthesize sugar. So oxaloacetate can be used to synthesize carbohydrates. Also, it can be used to synthesize certain amino acids, such as aspartate. So it is a good Lego with regards to that, and sometimes it is siphoned off of the TCA cycle for its work as a building block. Citrate can also be used as a building block, and we can understand that better when we think about the fact that citrate, when it goes backwards to oxaloacetate, of course gives off the acetyl-CoA again. So if we think about this kind of reversing engines sort of strategy for anabolism, the building block of fats and lipids is acetyl-CoA, and so citrate, we can think of that as being used in fatty acid and steroid synthesis. Of course, we recognize alpha-ketoglutarate as very, very similar to glutamate, and it takes very little anabolism to use alpha-ketoglutarate as the Lego to build up glutamate. So it can be an important precursor metabolite in the synthesis of amino acids, but also nucleotides. And then we can also finally recognize that succinyl-CoA is used to build porphyrins. And a lot of you will remember back to the protoporphyrin 9 ring and the porphyrin, the central porphyrin that we see in the hemoglobin and myoglobin molecules. So succinyl-CoA can be an important Lego in synthesizing those porphyrins. Now, I want you to think through this a bit. That is to say, if all of these intermediates are constantly getting pulled away from the TCA cycle, we need to think about how that affects the rate of the TCA cycle. So it's like these are siphoned off, they're like going down the drain. This is kind of what's going on here. Citrate's kind of swishing down a drain over here, or alpha-ketoglutarate down a drain over here. And essentially that's pulling away or siphoning away those intermediates. That means that's going to slow the rate of the TCA cycle. So it's all a balance, isn't it, between anabolism and catabolism. Now, on the flip side, there are not only reactions that siphon away intermediates of the TCA, there are also reactions that add in or fill up the TCA, and we have a special name for those reactions. They're called Anaplerotic reactions. I can't help but picture the TCA gas station every time I think of anaplerotic reactions. It's essentially reactions that allow one of the intermediates to get added get added in and fill up the whole cycle. And because it is that multi-step catalyst, if one intermediate gets replenished, then the whole cycle essentially gets replenished.